Okay, today we're going to talk about Punnett squares. So we learned a bit about heredity, and now we're going to learn how to calculate what that heredity could be. So a Punnett square is a tool used for understanding the patterns of heredity. It's a way that we can calculate what offspring we might get from two parents. It's a graphic used to predict the possible genotypes of the offspring, and from the genotypes, then we can figure out what it would look like, which would be a phenotype. How are Punnett squares used to predict patterns of heredity? Each parent has two alleles for a particular gene. An offspring receives one from every parent. So we're calculating the probability of how many different combinations we can get from these parents. Each compartment within the Punnett square shows an allele combination and the potential offspring. So here we have green seeds are recessive and yellow seeds are dominant. So green are recessive, yellow is dominant. So if we cross a green parent with a yellow parent, we can fill in this Punnett square. So here's our green parent crossed with our yellow parent. We're going to get one green allele and one yellow allele. One green and one yellow, one green and one yellow, one green and one yellow. So all of the offspring, if green is recessive and they all have one green and they all have one yellow, you're going to see all yellow offspring in this first cross. What color flowers are the offspring of purple and white flowered plants likely to have? Purple flowers and white flowers. They're not likely to have red because red was never introduced, and they're not likely to have yellow because yellow wasn't a part of a choice either. Okay, so let's look at alleles, alternative forms of genes. These are units that determine the hereditable traits. Easy for me to say. Dominant alleles, like capital T, remember we looked at this for tall pea plants. And then homozygous dominant. So that would be capital T, capital T. A recessive allele, a homozygous recessive would be two little t's, which would make a short pea plant. So if you have a heterozygous, where you get one tall from one parent and one short from the other, you will have a tall pea plant because it's dominant and it's going to mask the shortness. So let's look at phenotype ratios. So once we figure out a genotype, a Punnett square, we can calculate the percentage of offspring that will be of a certain genotype and a certain phenotype. So the ratio that we see, the outward appearance or the physical characteristics, is called the phenotypic ratio. So if it's tall, for example, a tall pea plant, we call it tall. If we're looking at the genotype, it would be big T something, either a big T or a small t. Or you could have the phenotype of a short pea plant, which would be two small t's. That's the only way you can get a short one. So the genotypic ratio is the ratio of the different genes in combination. So the arrangements of these genes and how they produce the different phenotypes. So here are some examples of genotypes you could get. Remember, genotypes is looking at genes. Phenotypes is the physical characteristic. So is it tall or is it short? That's a phenotype. A genotype is two big T's or big T, little t. So two big T's is homozygous dominant. A short pea plant would have to be homozygous recessive, which is little t, little t. And then if you want a tall pea plant that's heterozygous, it would be big T, little t. All right, so Punnett squares are used to show the possible combinations of the male and female sex cells, or the gametes. So let's look at a tall parent versus a short parent. Now, in all reality, it does not matter which parent you put in which direction. You're going to come up with the same answer every time. So don't get hung up in those details. So the first step in doing a Punnett square is lining up your parents. So one parent goes on the outside of the square here, and the other parent goes on the outside of the square here. So then we just bring one over. So this one has a big T to share, and a big T to share, and it has a big T to share here, and it also has a big T to share here. So now let's look at the next parent. Has a little t to share here and a little t to share here, and they would share a little t here and a little t here. So now let's put it all together. We have a big T from this parent and a little t from the other parent, a big T from this parent and a little t from this parent, a big T and a little t, a big T and a little t. So if we're going to look at the genotypic ratio, it's 100% big T, little t. Genes, right? What do the genes say? Big T, little t. It's also 100% phenotypic ratio of tall because every single one of these has a tall gene in it, doesn't it? A tall allele. So this produces the F1 generation. All the F1 offspring will be tall, and they will all be heterozygous tall, okay? F1, that's the first generation. Now let's cross the two F1s. This is exactly what we are going to do with our pea plants in our guiding question number 4. So we're going to simulate it here. So let's cross a big T little t with a big T little t, okay? So here we have a big T from this parent and a big T from this parent, a big T from this parent and a little t from this parent, a little t from this parent and a big T from this parent, and a little t from this parent and a little t from this parent. Now we have some genetic variability. We have a homozygous dominant and a homozygous recessive, and we have 2 heterozygous. So this is going to be tall, this is going to be tall, this is going to be tall. All of those have at least one big T. But you're 1/4 of the time, one time out of 4, right here's our 4, 25% of the time, you're going to get a small homozygous recessive pea plant. Okay, so here are the breakdowns. 1/4 will be big T, right here. See it? 25%, big T, big T. 1/2 will be big T, little t, because I've got 2/4. 2/4 is 1/2, or 50%. 2 divided by 4 times 100 is 50%. And then little t is 1/4, or 25%, 1 out of 4 chance that you will have a homozygous recessive. So your phenotype is 1 to 2 to 1, 1 to 2 to 1 genotype, and a 3 to 1 phenotype. 1, 2, 3 talls and 1 short. 3 tall, 1 short. 1 big T, 2 big T, 1 little t, that's our genotypic ratio, and 3 tall, 1 short is our phenotypic ratio. Okay, pedigrees. This goes right along with what we talked about in our Friday seminar with Kim Routsong and determining our family lineage, family trees. Pedigrees are a scientific tool, and we use them a little bit differently, but they tell us a lot of information to study patterns of inheritance. So we can look at a single trait and trace it through generations of a family. It can be useful tracing disorders, sex-linked disorders, like if it's something that affects males more, like color blindness, or disorders associated with one allele on a sex chromosome. We have, so a square, you have this square here, this is a male parent, and a circle is always the female. So a male parent with the trait, you've got part green, part yellow. This is the female parent without the trait. So the trait here is represented in green. Now, when these two organisms, maybe it's a pea plant, come together, you can have these offspring, okay? So here we have 2 males, one without the trait because dad gave this allele and mom gave this allele, both without the trait. And then you have a male offspring who doesn't get the trait from mom. She doesn't have it to give, but dad does and actually pass it on to that offspring. So then you can look at how the individual passed it down. Now this is a different setup than we just saw, but the same thing is true. If it's green, it's the trait that we're tracking. So this is a male that doesn't have the trait and a female that does have the trait. They have 2 offspring that are male, and they both have the trait. So this looks like mom has given this to both of these sons. Now this son then has offspring with a female that does not have the trait. And this shows you that the dad passed the trait on to a female, and the male didn't get it. So if we're looking at color blindness here, which individual has passed down the trait of color blindness to at least one offspring? you can see that this is colorblind, this is colorblind. They made 2 males that were colorblind because mom gave that trait to them. But a female to be colorblind would have had to have the trait both from mom and dad. So here we have, click the individual in the pedigree. There's where it started. How can a pedigree trace a trait through generations? Well, this can be useful in tracing inherited disorders such as cystic fibrosis. So let's look at this pedigree. Remember, a square is a male and a circle is a female, and they give you a key here. They also give you this key. Vertical lines connect children to their parents. So this means these 2 parents created this child. Or a solid square circle indicates that the person has the trait. So if it's a square, solid orange, that means that male has the trait. A circle, solid orange, that female has the trait. A half filled in circle indicates they're a character, but they don't display the trait. So if we look here, we have 2 carriers, and out of 5 children, they created 1 that has the trait, 2 that are carriers, and 2 that are not carriers and do not have the trait. Child 5, who is a carrier, then has offspring with a female that is not a carrier and does not display the trait. And they have 3 children, 1 which is a female that is a carrier. When that female then has offsprings with a male that's a carrier. So we've got 2 carriers here. You can do a Punnett square. If you did the Punnett square, you would see all this is true too. You have 2 of the 3, 1 male, 1 female, have the trait, and the 3rd is a carrier female. Okay, so let's look at, so this is a female parent being a carrier of hemophilia. So everywhere you see the dot, hemophilia is a genetic disorder in which your clotting mechanisms for your blood are very low or maybe not even existent. So if you get a cut, you could actually bleed out because your body doesn't have that coagulation to stop and make that clot up that scar, the blood that's coming out. So let's look at this possibility. So you have a male, XY is male. They are not a carrier. You have an XX, which is the female, and the female is a carrier. So let's look at what are the possibilities for their offspring. So 1X from dad, not a carrier. 1X from mom that is a carrier. 1X from mom that is a carrier. 1Y from dad, not a carrier. 1X from dad, 1X from mom, not carriers. 1XY from dad, 1X from mom, not carriers. So 50% of your offspring, 50% of your females will be carriers, 50% of your males will be carriers, but also 50% of the total will be carriers. Do you see this? XX is a female, carrier, non-carrier. And then XY carrier, XY non-carrier. All right, let's wrap up with a little bit of review. And I do have an Amoeba Sisters at the end that will help you understand Punnett squares a little better, and they are silly, so that makes it fun. A Punnett square predicts accurately the only genotype that will be produced. Is that true, or is that false? Pause and choose. The answer is false. It does help us. It is a tool to help us predict, but it is not the only way to accurately generate. An offspring receives 1 allele from each parent, true or false. Pause and put your answer. The answer is true, 1 from each parent. Big T, little t represents which of the following and why? Is it a homozygous dominant, homozygous recessive, heterozygous dominant, heterozygous recessive? Pause the video. The answer is heterozygous dominant. We have a big T, little t, and the big T is the dominant. So big T, little t tells us it's heterozygous because they're different, but it's dominant because the big T is present. Pedigrees can be useful for tracing sex-linked disorders. True or false? The answer is true. We just showed that with color blindness and sickle cell anemia. Okay, lastly, complete the Punnett squares for big T versus little t, big T. And then I want you to do that. Pause right now, do it, and then compare it to the one I'm about to show you. Pause right now.